Hi everyone, my name is Tom Keck, and I'm excited to tell you about the work my research laboratory performs at Rowan University. Our team is primarily interested in developing new medications for neuropsychiatric disorders, including schizophrenia, ADHD, pain, and substance use disorders like drug and alcohol addiction. Each of these disorders is different in many ways, but they share a common feature. They change how different parts of your brain talk to each other. We're trying to find new medications that might also affect how neurons in the brain talk to each other in the hopes that we may help patients with these conditions. To understand how we do this, you need to know just a little bit about how neurons in the brain talk to each other. Neurons send electrical signals that carry information, like how to move your arm or what chocolate smells like. When one neuron wants to pass that signal on to another neuron, they will often use a chemical signal to do so in a place called the synapse. Let's zoom in on the synapse and see how. When the first neuron wants to send a message to the second neuron, it will release chemicals called neurotransmitters onto its neighbor. Those create a new electrical signal in the second neuron. In many different disorders, this chemical message signal doesn't quite work right. So we're interested in finding new ways to affect that chemical signaling. We work with chemists that make unique molecules, like this one, for example, that we think could be helpful as medicines by changing how neurons talk to each other. Different chemicals can change how these neurons talk to each other, possibly increasing or decreasing the signaling between neurons at the synapse. In turn, this could help treat the symptoms or even the underlying causes of different disorders. And when we find medicines that work, we've partnered with chemists to design and test potential changes to those molecules to possibly make them work even better. We can take those new drug molecules and find out how those changes affect the interactions between the drug and the biological targets we're interested in. We will typically first test our new medications in special cells we grow in the lab, which allows us to measure how well our new compounds bind to our targets and what they do when they bind. Do they activate signaling within the cell, or do they block that signaling? If our new medications look interesting enough, the next step is to test them in animal models of neuropsychiatric disorders. We use models for diverse conditions, including schizophrenia, pain, and drug and alcohol addiction. And successful treatments in these animal models will hopefully lead to future testing in human patients. All of this research is possible due to the efforts of an enthusiastic team of talented and curious undergraduate and graduate students, each interested in developing new medications for neuropsychiatric disorders.